lab found a way to make an adult brain learn how to see things again. So I'm particularly interested in molecular cellular events that are responsible for that. So if you want to know more about it, please come talk to me after the lecture. So let's get back to the lecture. Oh, and please remember, feel free to stop me anytime if you have a question or comments. Okay, so today's topic is intracellular signaling. What's intracellular? What is a cell? Cell is a unit of life. So cells make tissue, organ, the whole body. So our body consists of many cells. And cells in our body know what's going on outside, because they communicate each other by sending signaling molecules. And they express different receptors for different signaling molecules. So once the signaling molecule binds to the receptor, that information will be sent inside the cell. And then ultimately, the cell will come up with some kind of response. And often the case, which means the signal goes to the nucleus and then turn on some genes and making some new proteins. So that is the intracellular signaling that we are going to learn today. And the key players in the intracellular signaling are proteins. What is a protein? Protein is a type of string, and 20 different things are attached to it. They are called amino acid residues. Yes. And depending on its nature, some amino acid residues are attracted to each other, and some are repellent to each other. And as a result, this string will fold into some shape. DNA, by the way, is also a type of string, but only 4 different things are attached to it. They are called
nitrogenous bases.
Nucleotides, yeah. And adenine, thymine, guanine, and cytosine are attracted to each other, and it makes double helix. And that's about it. Kind of boring. But proteins, you can make pretty much any shape with protein. All you have to do is come up with a sequence of how to line up 20 different amino acids. And then this string will fold into some shape by itself. Pretty neat, right? Another neat thing about protein is that just a little bit change in the local environment can trigger rather big change in the protein shape, which is called conformational change. And as you will see today, intracellular signaling takes full advantage of this protein conformational change. So there will be 3 important take-home messages today. First one, conformational change. So take any cell type, there will be an intracellular signaling going on, and they are pretty much the same. But the details are, of course, different. What kind of signal they get, what kind of receptors are expressed, and what kind of cell response they have. Details are, of course, different. And since this is a neuroscience class, we are going to focus on signaling in neurons. So roughly speaking, there are 3 ways to get signaling in neurons. One way to get it is to get it at the synaptic site. That's the first one. Yeah. And another way to get it is that the signaling molecule will be released and kind of diffused. And then the neighboring cells will get it. This type of signaling is called paracrine signaling. And the third way to get it is that the signaling molecule will be released somewhere far away in the body and then delivered through bloodstream, which is called endocrine signaling. Yes?
Sorry, can you explain paracrine one more time?
So paracrine means the signaling molecule is, once it's released, kind of diffuse.
Okay.
But it doesn't go far away as endocrine signaling. So paracrine affects neighboring cells, but not just very, very adjacent cells, which is pretty much touching to each other, like synaptic sites. Okay. Any other questions? Okay. So those are the ways to get signaling. So let's look at them one by one. First, synaptic signaling. So let's try to remember what we have learned so far. So the neuron has cell body in the center, and there are processes coming out from the soma, cell body. And there are several types of processes. And one type of process is specialized in sending output, which is called axon. And another type of process is specialized in getting input, and that is called dendrite. Yes. And within the neuron, this information will be transferred as an electric signal, right? So it goes very fast. But when it's ready to send that information to the next neuron, and then this electric signal will be converted to chemical signal. And the place that two neurons touch each other and communicate is called a synapse. Okay. So let's look at the most abundant type of synapse in the brain, which is glutamatergic synapse. And in this figure, upper side shows presynaptic terminal, which is at the axon, right? And the lower part is postsynaptic side. So the presynaptic side has, you can see, vesicles. They are called, anybody? Synaptic vesicles. On the synaptic vesicles, there is a protein called vesicular glutamate transporter. What it does is actively transport neurotransmitter glutamate into this synaptic vesicle. So it goes very, very high concentration, like a molar order. And when the electric signal arrives to this terminal, this synaptic vesicle will fuse to the plasma membrane and then release glutamate into this space between pre and post synapse, which is called, anybody?
Synaptic cleft.
Synaptic cleft, right. And on the postsynaptic side, there are receptors. And there are two types of receptors. One is called ionotropic receptor, and the other is called metabotropic receptor. AMPA receptor, shown here, belongs to ionotropic receptor. Ionotropic receptors are actually ion channel. So AMPA receptor is a sodium channel. So sodium ion concentration is higher outside or inside?
Outside.
Outside. Therefore, once the channel opens, sodium ion, which is Na plus charge, will move inside the cell. That's why it's depolarizing, and the chance of action potential will increase. It's excitatory. So it's excitatory synapse. Okay, so what happens into AMPA? Oh, wait. Before going to that, do you know why it's called AMPA receptor? Anybody know? Because substance called AMPA acts as an agonist for this kind of receptor. What is an agonist then? Agonist means it binds to receptor and activates it. What about antagonist? Antagonist binds to receptor and inhibits it. Okay. So how is this AMPA receptor working? So AMPA receptor is a sodium channel, but usually it's closed. However, upon ligand glutamate binding to this receptor, it triggers a conformational change in this protein. And then the channel opens, and sodium ion will flow in. So, as I just explained, sodium channel acts as an excitatory because sodium ion plus charge is higher outside the cell, and once it opens, it goes inside. Therefore, it will be depolarized. Chance of action potential will increase, right? Same thing can be said for calcium ion, because calcium ion is higher outside, and it's Ca2 plus, right? So plus charge will flow in. How about potassium? Potassium concentration is higher inside or outside?
Inside.
Inside. So once the channel opens, which is shown all the way on the right side, the potassium channel will flow out. Potassium ion is K plus, so plus charge will move outside. Therefore, it's hyperpolarizing. So the chance of action potential will decrease, so it's inhibitory. Yes?
Are you saying the AMPA is permeable to both sodium and calcium ions?
Oh, no, this was just different channel will make a different action.
Okay.
Either.
Okay.
Okay, so the
it's just sodium.
question was, is AMPA acts for sodium and calcium? Well, let me answer your question, actually. AMPA has different types of subunit, and depending on the subunit composition, it could act as a calcium channel too. So yes and no. It depends on the subunit composition. But main point here is, depending on the channel, because of the ion concentration, action of channel is quite different. It could be excitatory or inhibitory. Now, let me ask you a question. What is the difference between a pump and a channel? Yes?
Channel only uses diffusion, which is not active, passive, but pump is actively making the ions to move one direction.
Perfect. Perfect answer. So, difference is pump actively transport ion using energy, whereas channel is passive. So depending on the concentration of the ions, it could move either way. Okay. Here comes the tricky one. chloride channel. Chloride gradient changes dramatically during the development. In a young neuron, chloride ion is higher inside the cell. Chloride ion is CO minus, and once the channel opens, minus charge will move out. Therefore, it's depolarizing, so it's excitatory. And at around birth, this gradient flips. Now, in a mature neuron, chloride ion is actually higher outside than inside. So the same channel opens, and now it flows the opposite way. So the minus charge goes in. Therefore, it's hyperpolarizing. So it becomes excitatory to inhibitory. It goes through dramatic change during the development. Okay, so let's go back to a glutamatergic synapse. So we were looking at ionotropic receptor, which is AMPA. But like I said, there's another type of receptor, which is called metabotropic receptor. At glutamatergic synapse, there is a metabotropic glutamate receptor, mGlu-R, this one. So how does this work? So metabotropic receptors are type of G protein coupled receptors. What is a G protein then? Well, G protein prefers to bind guanylate phosphate. Usually, it likes to bind GDP. But upon a ligand binding to the receptor, this receptor changes conformation. And that information will transfer to G protein. And now, G protein prefers to bind GTP instead of GDP. And this action will cause conformational change in the G protein. Actually, G protein consists of 3 subunits, alpha, beta, gamma, of which alpha subunit will come off and then goes to bind some other protein and changes conformation there. So that's how you send signal. So there are several types of G proteins, and in the case of mGlu-R, it's linked to GQ. What GQ type of G protein does is to activate a type of enzyme called phospholipase C, or PLC. There's another type of G protein called GS. For example, norepinephrine will bind to beta adrenergic receptor, which is linked to GS. And what it does is it activates adenylyl cyclase. And the third type is GI. For example, dopamine binds to dopamine D2 receptor, which is linked to GI. What it does is, instead of activating, inhibiting adenylyl cyclase. So as you can see, what G proteins do is basically manipulating some kind of enzyme, right? So what is an enzyme? Enzyme catalyze some chemical reaction. So once the enzyme is activated, some chemical reaction will go on. So as long as the enzyme is active, something is kept produced. So it's a nice way to amplify the signal. So this is the second important point today. Intracellular signaling takes advantage of enzyme activation to amplify the signal. And then those enzymes, what do they produce? In the case of adenylyl cyclase, it produces cyclic AMP. And phospholipase C, PLC, will produce two products. One is diacylglycerol, or DAG. Another is inositol triphosphate, PI3. I'm sorry, IP3. Confused. So these molecules, the product of some chemical reaction, is called second messengers, because the first one is, of course, these neurotransmitters. And this information will be converted to different messengers. So we call it second messengers. And this second messenger will go on to transfer that information to do something. What do they do? Well, cyclic AMP will activate protein kinase A. Diacylglycerol, or DAG, will activate protein kinase C. And I will come back to IP3 later. But as you can see, they activate something called protein kinase something. So what is protein kinase? Protein kinase is a type of enzyme and which mediates phosphorylation of a protein. So phosphor group is PO4 3 minus. So once it's phosphorylated, the local environment of that protein will be changed. And as a result, it will cause conformational change. And that's how you send this in. Now, I'd like to think about this system. If you have some switch, on switch, you might want to have off switch too, right? Because otherwise, once the fourth signal is gone, you can't know what's happening. Or maybe it was gone for a while and then it came back, and you can't respond to that. So you need to update the information. Well, there is off switch, protein phosphatase. What it does is dephosphorylates the phosphoprotein. So this is the third important point. Intracellular signaling has both on and off switch, because it's important to keep updating, right? So let's think about other, we already saw a lot of signaling going on from neurotransmitter to receptor, and then PLC, and then second messenger, and then PKC. So they all have to be upgraded. So how do you turn off? Let's think about here, neurotransmitter to receptor. How do you turn off? Well, this interaction is not covalent, so it's just equivalent. Equilibrium is influenced by concentration. So if the neurotransmitter concentration goes down, and then signal will be gone. So you might want to remove the neurotransmitter from the synaptic cleft quickly, so you can respond, you can get the updates. And do you know who is doing that job? Glias, yes, especially astrocytes. So astrocytes has a very important role in the synaptic transmission. It removes, take away glutamate from the synaptic cleft. Okay, so we've been looking at this signaling, and how is this protein kinase C, PKC, work? Well, PKC has two domains. One domain is called catalytic domain. That's where you do this catalytic action. And there's another domain called regulatory domain. And usually, regulatory domain is hiding the catalytic domain. So this molecule is usually inactive. However, when the messenger, secondary messenger, DAG comes to bind it, and then it changes conformation. And now it goes to inner layer of plasma membrane and bind it, and as a result, catalytic domain will be exposed, and now it's active. So that's how it works. Well, how about a GX? So as we learned, adeninocyclase will make cyclic MP, and cyclic MP in turn activates protein kinase A, right? How does this work? Protein kinase A, PKA. Well, it's actually a very similar story as PKC. So PKA also has two domains, catalytic and regulatory domains. And regulatory domains is hiding catalytic domain, so it's usually inactive. And upon the ligand cyclic MP binding, it triggers conformational change, and now catalytic domain is exposed and it's active. So you get the pattern, right? Okay. So that was the glutamate synapse, glutamatergic synapse. What about other types of synapse? Let's look at acetylcholine synapse, cholinergic synapse, because acetylcholine, cholinergic neurons are abundant in the peripheral nervous system. So what kind of receptors do they have? Well, cholinergic synapse also has ionotropic and metabotropic receptors. Nicotinic acetylcholine receptor belongs to ionotropic receptor. It's also sodium channel. Therefore, it's depolarizing once it opens, so it's excited. That's why cholinergic synapse is excitatory. The reason it's called nicotinic acetylcholine receptor is yes, because nicotine acts as an agonist for this type of receptor. Now, nicotine is abundant in tobacco. Do you smoke? No? If you're thinking of smoking, I wouldn't recommend that. So, like I said, there are lots of cholinergic neurons in the peripheral nervous system, but there are a small number of cholinergic neurons in the brain. And one of the very important places that these neurons project to is the center for reward system. Once you take nicotine, that reward system will be activated. Well, in fact, it will hijack the reward system. That's how you get addicted. So I wouldn't recommend that. Okay, so that's the ionotropic receptor, and it also has metabotropic receptor, which is called muscarinic acetylcholine receptor. And as you can imagine, the reason it's called muscarinic is because a substance called muscarine acts as an agonist for this type of receptor. Muscarine is extracted from some type of mushroom. Okay, so that's the acetylcholine receptors. How about GABA? Gamma aminobutyric acid. Well, it turned out GABAergic synapse also has inotropic and metabotropic, very similar stories. GABA A belongs to inotropic receptor, and it's a chloride ion channel. So earlier, as I showed you, chloride ion is very tricky. In young neuron, it's excitatory. However, in mature neuron, at around the birth, it flips. In mature neuron, chloride ion is higher, so once it opens, it goes inside. So the minus charge will flow in, therefore it's hyperpolarizing. That's why GABAergic synapse is inhibited. Okay, and they also have metabotropic receptor. For example, GABA B receptor is a metabotropic receptor. So it's a G protein coupled receptors. But this guy is a little bit different from others. Once ligand GABA binds to the receptor, it changes conformation, and then it causes conformation in the G protein. However, in this case, instead of alpha to come off, beta gamma will come off. And then it goes to bind to a type of potassium channel called GARC. And as we looked at earlier, potassium channel is inhibited, right? So, because potassium ion is higher inside, so once the channel opens, it flows out, and potassium is K plus. Plus charge will move out, where it's hyperpolarized, inhibitory. So they are both inhibitory. However, there is a critical difference. In the case of GABA A, itself is a channel. So once the GABA binds to it, and then it changes conformation of GABA A, and then ion channel opens. So the action is very fast. On the other hand, GABA B is a G protein couple, so it binds to receptor, changes conformation, and then that will cause conformational change of G protein, and then beta gamma subunit will come off and then go to GARC and binds to it, and that triggers another conformational change in the GARC, and finally, potassium channel opens. So it takes time. And similar thing can be said to other type of synapse. Usually, ionotropic action is very quick. Metabotropic is slow. Okay. Now,
So if ionotropic's quick, what's the purpose of having it slow?
slow. Okay, the question was, why do you want slow responding receptor? Why this isn't enough? That's a very interesting question, because you might want to have long inhibition sometimes. This way, you could make a population of cells operating in a certain way, and you can make interesting rhythm. So it's very important for, if you think about population of neurons activity, this way is very useful. But other than that, there are a lot of action that metabotropic receptors do, not just inhibiting, but sending signal inside too. Okay. Any other question? Yes?
Does that mean metabotropic have a long-term effect compared to ionotropic?
Yes, you can say, so the question is, metabotropic receptor has a long-term effect compared to ionotropic. You could say that the whole thing is very slow. Yes. So ionotropic receptor usually sends electric signal, so that action is very fast. However, that could trigger some other signaling cascade, which could act pretty long term. Yes, another question.
So if GABA binds both, if the presynaptic neuron releases GABA, does that mean that both ionotropic and metabotropic are gonna be activated, or does that mean, can you differentiate which one gets activated?
So the question is, so if you have both GABA A and B on the same synapse, and then are they both activated at the same time, or do they have different role? Well, basically the answer is depending on the neurons. And sometimes GABA B is only expressed, it's actually it could be presynaptic terminal too, because once it's released, it could be binding to the presynaptic site too. So they might be expressed in a different place, and action will be independent. And yeah, that's the quick answer. Yes. Okay. All right. So can I go back to glutamatergic synapse then? Because I skipped one important thing. I haven't thought about it. Something very important, which is this. NMDA receptor, N-methyl D-aspartate, acts as an agonist for this receptor. And it's also a type of ionotropic receptor. It's actually calcium channel. But the way this receptor works is kind of tricky, because usually magnesium ion blocks the pore so the calcium can't get in. So only when the magnesium ion is gone, then you can see this channel. So how to get rid of the magnesium? One way to get rid of it is to depolarize the membrane, and that would cause the conformational change. And the reason I said it's a very important receptor is that this calcium is used as a signaling molecule. So, to be able to use calcium ion as a signaling molecule, neurons has to get rid of this calcium ion from the cytosol. So using pump, it gets rid of calcium ion from this space cytosol. So the concentration of calcium ion in cytosol is extremely low. And there are two ways to get calcium signaling. One way is to get it from outside through the plasma membrane. And as I just explained, NMDA receptor, which is a type of ligand-gated calcium receptor, can do that. Another way is to get it is this voltage-gated calcium channel. So these guys will open only when the membrane is depolarized. So these are the way these guys can make the calcium flow in from outside. Another way to get the calcium is get it from endoplasmic reticulum, ER. So ER stores calcium ion. And on the ER membrane, there is a ligand-gated calcium channel. What is a ligand? It's actually IP3. So I told you I will come back to it later. Here comes. IP3 actually binds to this receptor, and then calcium will be released from ER. So these are the way to get calcium signaling going on. So how does this translate into another signal? It's like, what can calcium do? Well, actually, there is a kinase which can be activated by calcium called CAM kinase. And the way CAM kinase works, you will see it's very familiar story now. Well, it has catalytic domain and a regulatory domain. Regulatory domain is hiding catalytic domain, so it's usually inactive. However, when the calcium is in, it binds to a protein called calmodulin. It likes to bind calmodulin, and this calcium calmodulin complex will bind to regulatory domain, and then now conformation will change and catalytic domain will open, and it's active. So, like I said, neuron uses calcium as a very important signaling molecule. So they have special structure. If you look at the dendrite, so the dendrite is where you get input, right? If you look at this dendrite, the process, you see this protrusion, lots of protrusion in the dendrite. These are called dendritic spine. And the spine has mushroom-like head, and usually it has neck, narrow point. So by having this type of shape, you can compartmentalize. So once the calcium flows inside this balloon-like thing, and then just a little bit amount of calcium can make a lot of concentration change because it doesn't diffuse as well. So it's very effective way to get calcium signaling going on. And at the tip of the mushroom head, you see this blue structure. It is called postsynaptic density. So it's actually, this is the postsynaptic site. Why is it called postsynaptic density? Because series of proteins are densely packed around this region. What are they? Well, they have receptors and all kinds of kinase, other molecules, signaling molecules are linked with this protein. As you can see, mGluR is linked to ER. So once the neurotransmitter or a signaling molecule arrives, and then everything is right there, ready to do action. So it's very effective way to send signal. So that's postsynaptic density. And if you look at electron microscope image, postsynaptic density, you will see why it's called postsynaptic density. It's really, really dense, dark portion you can see. Okay, so this was the synaptic signaling. So let's move on to the next one, which is paracrine signaling. Okay. The most well-known paracrine signal in neuron is neurotrophic factors. For example, nerve growth factor, NGF, is a type of neurotrophic. And it has a receptor called TrkA. This TrkA receptor belongs to type of receptor called receptor tyrosine kinase. Why is it called receptor tyrosine kinase? Well, its cytosolic part is actually enzyme kinase, tyrosine kinase. And upon ligand binding, these molecules, the receptor, will dimerize. And then they will phosphorylate each other. There are tyrosine residues in both sides, and they will phosphorylate each other. And once they're phosphorylated, that would trigger series of signaling shown here. So it can do multiple things all at once. For example, it can activate PI3 kinase, type of kinase, which in turn activates another kinase called AKT, and they send signal. Another one, RAS. RAS is a type of G protein. However, instead of having alpha, beta, gamma, it's just one protein. So it's called small G protein. And what it does is activate series of kinase. In the end, it activates something called MAP kinase. So that's also one pathway. And also it activates PLC pathway. So it can produce DAG and AP3. So what neurotrophin does is basically it's very important signal for a young growing neuron. It supports cell survival, and it supports also promotes growth and maturation. So I want to show you what a neurotrophin can do. So here's an example. This is another type of neurotrophin called brain-derived neurotrophic factor, BDNF. And as shown here, it's the tip of axon of young neuron. And as you can see, there's some structure here. Do you know what this is called? Okay. Well, this is called growth cone. So it's a hand-like structure, and it's very mobile and seeking for cues, both attractive cue and repellent cue, and makes decision of which way to grow. And now you can see at the right top corner, there's a glass pipette, and we will apply BDNF from here. And this is time 0, 20 minutes, 40 minutes, 60, 80. So you can see it's clearly attracted to BDNF, right? So it promotes axonal growth. Well, I would like to show another example of what neurotrophin can do, which is that BDNF can enhance communication between neurons. So I'm going to show you some video, and it's a calcium imaging. So in calcium imaging, you use calcium dye. Calcium dye is a fluorescent molecule, but it fluoresces only when the calcium binds to it. And as you can see, there are a lot of dots here. These are the soma of the neurons. So you can see a lot of neurons here. And I will show you 2 videos, before and after adding BDNF. So I will show you the before video first, but I have to warn you, these neurons are not really active. So if you pay attention to somewhere here and somewhere here, then you will see something. Okay. So that's before BDNF. And the same set of cells on the right side, you can see it's the same cell. And just 5 minutes after adding BDNF. Let me show you once again. So before is like this. And after. I hope I can convince you that it has a tremendous effect. Because I did this experiment. Okay, so. Okay. So, we were looking at so many different signaling pathways, but in the end, what do they do? And as I told you, that often the case it goes to a nucleus and turns on some genes. And a good example is this molecule called CREB. So, scientists noticed that if you look at that gene, there's some sequence. And if you have this particular sequence, it seems to be this gene will respond to cyclic AMP. So they named that sequence cyclic AMP responsive element, CRE. And then later on, they found that there's a protein binds to it. So they named it cyclic AMP, CRE binding protein, CREB. And then later, they found this CREB can be phosphorylated by pKa. So cyclic AMP will activate pKa, and then this pKa will phosphorylate CREB. And once CREB is phosphorylated, it goes to the nucleus and binds CRE, and then turn on those genes. It turned out it's not just pKa, other things can phosphorylate CREB too. For example, if you get calcium signaling, and then CAM kinase can also phosphorylate this CREB. And the neurotrophin can activate RAS, and then MAP kinase can also phosphorylate. So many things could turn on this CREB. So what kind of genes will be expressed? Well, genes that are important for growth or survival. Okay, so any questions? Okay. So let's go to the last one, the third one, endocrine signaling. Okay. So the typical endocrine signal is a hormone signal. For example, stress hormone, glucocorticoid. Glucocorticoid is pretty hydrophobic, so it crosses plasma membrane. So the receptor doesn't have to be waiting on the membrane. It's actually in the cytosol. And ligand crosses plasma membrane and then binds to it. And then it triggers conformational change in the receptor. As a result, this receptor itself will go into nucleus and bind DNA. And then turn on the gene. So it does everything by itself. And as you can imagine, because you have to do everything by yourself, it's kind of slow. But, well, hormone itself is coming far away from the body, so when it arrives, it's already kind of slow. So it doesn't have to be very fast, right? So, stress hormone is released when you are under stress. Why do we have something like that? Well, if you are exposed to stress hormone briefly, it's actually good for your neuron and it enhances learning, which sort of makes sense because if you encounter something dangerous or scary, you might want to remember it so you can avoid it next time, right? So it makes sense. But if you are exposed to stress hormone chronically for a long time, oh, that's a bad news. It will damage your neurons. So there's an animal study, mouse study, that if you separate newborn pups from mother, and then those pups get very, very stressed. And that would affect tremendously the development of the brain. And it will affect behavior. It will be stressed and depressed. So it's a very serious consequence. So, important lesson, don't get stressed too much. So, okay, so any questions? Yes?
When you say slow, does that mean by comparing to other pathways? Like how slow it is?
Okay, so when you say slow compared to other pathways, yes. So, for example, we are looking at synaptic signaling, which is very fast, although it's slower than electric signal. So it kind of slows down, but it's still very fast. But hormone is released somewhere far away. You know, it's not even in the brain. It could be somewhere else. So then it has to arrive through bloodstream, and then you get this action which is not designed to have very fast, quick. So it's relatively slow. Yeah. Any other question? Okay. So remember our 3 important take-home messages today. So first one is conformational change. So protein conformational change is the key for sending signal one to other. And the second one is using enzyme to amplify signal. Right. And the third one is that
On and off switch.
yes, you have to have pretty much on and off switch, so you can update this information. Okay. All right. So if there's no other questions, that's all. Right.